This video is about the function of T cytotoxic and T helper cells. I'll start by just saying a little bit about T cells. They originate in the bone marrow but mature in the thymus. They have T cell receptors, and T cell receptors can only recognize processed antigen that is bound to major histocompatibility complex molecules, or MHCs. The first kind of T cell I'll talk about is the T cytotoxic cells, or TC cells. They recognize antigen presented by MHCs1. They kill that cell directly. In order to do that, contact between the TC and target cell is required. Once it makes contacts, its granules move to the contact site. Degranulation occurs and causes pores in the target cell membrane, so it falls apart and dies. It also includes granzymes in its granules, which cause apoptosis, or programmed cell death. This diagram kind of shows, here's your TC cell. It has a CD8 co-receptor. Here's its MHC1 proteins. There's the T cell receptor binding to your antigen. So once it makes contact with the target cell bound to MHC1, it releases its granules, which causes the target cell to die by apoptosis. So the key things to remember is that the T cytotoxic cells use a CD8 co-receptor and only can recognize MHC1 proteins. T helper cells are the other kind. They interact with MHC2 proteins, and they work by releasing cytokines to promote immune responses. There are many different kinds of T helper cells. The first one I'll mention is Th1 cells, which initiate immunity and inflammation by activating macrophages by secreting cytokines. The cytokines that they use is interferon gamma, tumor necrosis factor alpha, and granulocyte monocyte colony stimulating factor. They're activated by antigens presented by macrophages in the context of MHC2 proteins. Activated cells produce cytokines that stimulate the macrophage to increase phagocytic activity and promotes inflammation. Once a macrophage is activated, it's much more efficient than it normally would be. Th1 cells focus on intracellular bacteria and tumor cells. It's able to target tumor cells because tumor cells, even though they are the host own cells and made by the host body, tumors still express specific tumor antigens which can be targeted for destruction. Th1 cells also play a role in rejection of transplanted organs. So that's why people have to be on so much medicine after they have organ transplants. This diagram shows how Th1 cells activate macrophages. It's pretty similar to the last one, except here is a CD4 receptor, and it uses MHC2 proteins. There's also more cytokines involved, and of course it's a macrophage. The Th1 cell binds to the macrophage that's presented by MHC2 proteins, and once they come in contact, it releases its granules. It releases its cytokines, which cause the macrophage to release its own cytokines, which then result in increased phagocytotic abilities and inflammation in the infected sites. So the key things to remember about Th1 cells is that they target intracellular pathogens, use CD4, and can read MHC2 proteins. So Th2 helper cells regulate immune response to extracellular pathogens. They stimulate antigen-reactive B cells to make antibodies. B cells are usually covered with antibodies that are antigen receptors. Antigens bind to the B cell receptors. B cells don't automatically make antibodies. Antibody-bound antigen is taken into a B cell by endocytosis and then it is degraded. Peptides from the degraded antigen is presented on a B cell MHC2 proteins. The B cell then presents the MHC2 protein to the Th2 cell. Then the Th2 cell secretes interleukin 2 and interleukin 5, which are the cytokines that activate the B cell. And then the activated B cell differentiates into a plasma cell, which makes antibodies. Here's a diagram of how that happens. This one's a little bit different from the last two because it's B cells that further differentiate where the other ones just were activated. So this one has a little more changes. The Th2 cell binds to the B cell via MHC2 proteins and the T cell receptors. There's the antigen in the middle. The B cell can then become a memory cell, which stays in the immune system for a long time, or it can become a plasma cell that secretes antibodies. During the secondary response, a memory cell can then become a plasma cell later. So it's much faster the second time if you get the same pathogen. Okay, sorry. Then another kind of Th cell is Th17 cells. They're triggered by antigen interactions, and they recruit neutrophils. Undifferentiated Th cells turn into Th17 cells because of dendritic cells. So this one has neutrophils and dendritic cells involved. When the dendritic cell encounters a pathogen, they present the antigen and secrete interleukin 6 and transforming growth factor beta, which cause naive Th cells to turn into Th17 cells. Once they turn into Th17 cells, they secrete interleukin 17, which is where that gets its name. Interleukin 17 causes other cells to produce cytokines that also attract neutrophils to the site of infection. This amplifies innate immunity. Okay, so here's how that one works. You can see here's your dendritic cell, and here's your neutrophil. The dendritic cell has an antigen bound by PRR, a pattern recognition receptor. Here's the MHC2 proteins. Once your Th17 cell binds to your dendritic cell, which has an antigen on it, the dendritic cell releases interleukin 6 and the growth factor beta. The Th17 cell secretes interleukin 17 into the tissue cells, which then secrete their own cytokines such as CXCL8, which then calls neutrophils to the site of infection.